Okay, so this poster is about protein sequence similarity. So here in this research, we introduced a new method called CB search, and this is for searching for similar motifs with biased compositions. So what are similar motifs with biased compositions? Here we have several examples. This is collagen-like domain, this is transmembrane domain, and here we have RGG box. So you can notice that all of these regions are rich in one or a few residue types. So there might be a question why searching for these motifs needs another method. So the answer is that most of the state-of-the-art methods like BLAST and HH masked these regions or decreased their significance in order to improve homology search, right? So therefore, these motifs are largely unexplored and belong to so-called dark proteome. Okay, so here we introduced similarity score. This is basically the alignment score over query self-alignment score. So we compare the alignment score to the perfect alignment, right? So in comparison to e-value, this score is length independent. So there may be a question whether this longer collagen-like domain is more biologically significant than this shortened transmembrane domain. From e-value point of view, yes. However, from similarity score point of view, it is not. So this similarity score also normalizes logarithm base, which were used to calculate scoring matrices, because different scoring matrices use different logarithm base. Therefore, in e-value, developers introduced such a lambda parameter, which normalizes this logarithm to natural logarithm. However, in the case of similarity score, we utilize change of base rule, and the final score is independent of this intermediate base C. Okay, so then we have also semicon method, which identifies regions with a similar composition. So we can see that for this query, similar composition has such a sequence. This sequence, they are very similar. Here in this sequence, this asparagine homopolymer is before homopolymer of glutamine. This is a similar sequence. And here we have also short tandem repeats of asparagine and glutamine. So these hits have similar compositions, however, these amino acids are arranged differently, right? So for instance, here we have short tandem repeats, and here we have two homopolymers. Next, we also introduced three alignment strategies. So this is local alignment, and the drawback is that if we have fused compositionally biased regions. So the first part is homopolymer of glutamine and the second homopolymer of arginine. Then this local alignment frequently only take a single part into account when aligning both sequences. In the case of global local alignment, if the motif is shorter than the query motif, then we apply here gap and extend penalties. So in the case of global alignment, then we take this query and simply perform global alignment on these hits, and then we can check whether the sequence is shorter or longer, or maybe the residues are arranged differently. Here we have also results for our analysis, and we can see that CB search in three variants with local, global, local, and global alignment outperformed BLAST method in the case of transmembranes. In the case of RGG boxes, it performed a little bit better. And in the case of keratin type 2 head, it also had better results. So the results are better, however, we didn't notice a significant difference between alignment algorithm. For the transmembranes, they are very similar. For RGG Gbox's global alignment with semicomp performs a little bit better over here. However, the difference is still not significant. Maybe here it performed a little bit better, but the difference is not astonishing as in the case of similarity score. It means the alignment algorithm doesn't have such a huge impact. So just to conclude, our method improves annotation retrieval. So if you have a query motif, then you might search for similarities, and most of the annotations which are assigned to this found motifs should be relevant to the query. Global alignment with semicomp and global and local alignment include all parts of the query sequence more frequently than local alignment. This is because here we apply gap open and gap extend penalties. And finally, our similarity score introduced in the study outperforms e-value, which is visible over here, so the results are significantly better. If you want more details, then you can head into the scientific article that was published in BMC Bioinformatics, or if you want to try out the method, then you can go to GitHub page, download the code, and try it out. Thank you very much for your attention.